All right, we are going to continue our study of chromosomes and genetics with this next little mini vocabulary lesson. So I'm going to do some guided notes. You write what I write. So we have chromosomes. Remember, chromosomes are coiled up and organized DNA. DNA is what they are made of, and chromosomes are, once they're kind of, we represent them often something like this, like these little chromosomes. You'll see these in a picture. So for humans have 22 pair of autosomal chromosomes. That means these are, they're kind of matching ones, one from each parent. So we have 22 autosomal ones, and we have the 23rd pair is going to be our sex chromosomes. And there's really just one pair of those, right? Whoops, one pair. So the sex chromosomes are these XX for female and XY for male. So the mother is donating, if you will, the X chromosome to the offspring, and the father, since the father has a Y to give, can donate either an X that he has or a Y. So the gender of an offspring comes from the father, in humans anyway, but that's not the same in every animal. So now let's talk a little bit more, let's go a little deeper. In a chromosome, we can have a little section of a chromosome which will code for a particular trait. So a section of DNA that codes for a specific trait is a gene. Codes for a specific trait is a gene. Right, so that's our another big vocabulary word. So it's just a section of the DNA, and it could be like a gene for a particular thing, and it's going to be a section on the chromosome itself. Now you can see we have two chromosomes of the same thing, so you would have two genes for, we're going to use eye color, for example. So if you have two separate genes for eye color, they might be the same or they might be different. And so the genotype of an organism is the actual it's the actual genes that determines the phenotype. We'll get to that. It's actually the set of genes that organism has for, and we're usually talking about a particular trait. So the set of genes for a trait. So an example is, well, we'll get to example 2, but an example would be if you're talking about eyes, color, or whatever trait you might have, it would be like the letters that we use to represent the allele. So big V, little b, dominant, recessive. We're going to get to those as well. So the phenotype are the observable characteristics that are displayed by the organism. So it's the observable characteristics displayed by the organism. So in this case, you would say like the phenotype would not be dominant recessive. The phenotype would be, example, brown eyes. Or example might be tongue rolling, an ability that an organism has. So Now you're saying, what is this BB business? If you have two forms of the same gene, then those are two different, we call those alleles. I'll write that down here a little bit later. So we have two different kinds of traits. We have dominant traits. So for traits, we have dominant, and they are present, they're kind of the stronger ones. And they can be present in the phenotype, so we will see them show up if only one allele is present. And then recessive, they're not quite as strong, and they only show up in the phenotype when the dominant is not present. So dominant is usually represented by a capital letter, so a B, for example. And we use other letters as well. I'm just using B for now. So a capital letter represents our dominant traits, and our recessive traits only show in the phenotype when no dominant allele is present. So that would be, for example, BB. So if you have if both versions are a little b and a little b, then that would be recessive. So they're going to show up BB if it's like that. But for dominant, you're going to see the dominant if it's BB, like that, or like this. So you're going to see the dominant trait even if only one of those dominant alleles is present. So an allele, let's define that now. An allele is a variety or a version, I'm going to say version, of a specific gene. And we usually just talk about one gene at a time. Sometimes we'll look at two things at once, but so it's a variety or a version of a specific gene, and it's at a specific location. And in genes, when we're talking about a specific location, we call it a locus. You can see it's the same root word as location. So that's location on the chromosome, right? Now, how do we determine what kind of offspring we're going to get? So to determine that, we use something for simple inheritance. We use something called a Punnett square. So to determine the offspring, we use a Punnett square, and it's 2 N's and 2 T's. A Punnett square, okay? And a Punnett square allows us to take, the two genes that the parent might be able to donate from each parent and see how they might independently mix up. So we do it like this. If we're talking about one set of traits, then we would say we have a parent that has brown eyes. So for eye color, we're going to say this is brown eyes, and that's dominant over a little d, which is blue eyes. Now, in reality, eye color is a little bit more complex. It can be more than just one set of genes, but brown and blue work a little bit like this. When something works on a Punnett square in just a very straightforward single gene, dominant recessive kind of thing, then we call that that follows Mendelian genetic rules. So if we have this parent that's BB, and say we have another parent that's also BB, then we would put one parent on the top here, BB, and one parent on this way down the side, B there and B there. So basically you're going to have, like, these are the genes from one parent, right? And then these are the gene from another parent. So that's where those are coming from. You're going to take one parent and put it on the top, one parent on the side. And it doesn't really matter which one is which. And then you're just going to put them together. So in the middle, if you got this allele and this allele, the offspring would be this. Again, I'm going to take this big B, and we usually just put the big B first, right? So big B, little b here, B, B. Here I've got one big and one little. One big and one little, and over here I have one little and another little. So I have dominant, 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 recessive, two versions of that, and one dominant, recessive, recessive. Now, the thing to remember is that these are the percent chance. This tells us not exactly what the offspring is going to be, but it's going to tell us the percent chance that we might end up with any one of these genotypes. So we have for phenotypes, we have a 75% chance of the dominant phenotype. Because remember, since all three of these are dominant, even though these are dominant recessive, they would all show up as brown eyes. And there would only be a 25% chance of those blue eyes of the recessive. Now there's one more set of words that I want to make sure that you've got, and that is homozygous and heterozygous. And these, again, you've seen these root words before. Homo means same, hetero means different. So that's going to make it really easy for you, because homozygous means that you have two of the same alleles. So you could have two dominant or two recessive. Those would be homozygous. So you could say if you had something that's homozygous dominant, then you would know it's this. If it's homozygous recessive, you would know it's this. If it's heterozygous, hetero means different, then you're going to have one dominant, one recessive. And those could be B's or T's or whatever. So that's just a little introduction to some of the vocabulary that we use when talking about Mendelian genetics.